Hi guys, we are going to see about physiology of red blood cells in detail. At the end of the video we know about the normal value, morphology, size and lifespan, properties, fate, function, variations in number, variations in size, variations in shape of red blood cells. Introduction. Red blood cells, RBC, also known as erythrocytes, are non-nucleated formed elements in the blood. The red color of RBC are due to presence of hemoglobin. Normal value. Normal RBC count ranges between 4 and 5.5 millions per cubic millimeters of blood. In adult male 5 millions per cubic millimeters. In adult female 4.5 millions per cubic millimeters. Morphology of red blood cells. RBC are disc-shaped and biconcave, dumbbell-shaped. The central portion is thin. The periphery is thicker. The biconcave contour of RBC has some mechanical and functional advantage. It's non-nucleated cell because of the absence of nucleus. DNA is also absent. Other organelles such as mitochondriae and Golgi apparatus are also absent. Since mitochondriae is absent, energy is produced from glycolytic process. Advantages of biconcave shape of RBC. Its help in equal and rapid diffusion of oxygen and other substance into the interior of cell. Large surface area is provided for absorption or removal of different substance. Minimal tension is offered on the membrane. When the volume of cell alters. While passing through capillaries, RBC can squeeze through capillaries without getting damaged. Size and lifespan. Diameter, 7.2 micrometer, 6.9 to 7.4 micrometer. Thickness. At periphery the thickness is 2.2 micrometer and the center is thinner with 1 micrometer. The difference because of the biconcave shape. Surface area, 120 square micrometer. Volume, 85 to 90 cubic meter. Average lifespan of RBC are 120 days. After that time the old RBC are destroyed by reticulo endothelial system. Properties of RBC. Rolex formation. When blood is taken from blood vessels the RBCs are piled up one above another like a pile of coins. This property of RBC is called Rolex formation. It is accelerated by plasma proteins that are globulin and fibrinogen. Specific gravity of RBC is 1.092 to 1.101. Packed cell volume, PCV. The PCV volume of RBC are expressed in percentage. It's also called hematocrit value. 45% of blood and 55% of plasma volume. Suspension stability. During circulation the RBC is suspended or dispersed uniformly in the blood. This property of RBC is called suspension stability. Fate of RBC. When RBC becomes older, 120 days, they become fragile. So, they are destroyed while squeezed through capillaries which has lesser or equal diameter of RBC. The destruction mainly occur in spleen because they have small capillaries. So it's called graveyard of RBC. Daily 10% of RBC are destroyed in young adults. It causes release of about 0.6 gram per deciliter of hemoglobin into plasma. From this 0.91 to 1.5 mg per deciliter bilirubin is formed. Destruction of RBC. Release of hemoglobin. Iron plus ferrite, stored and reused. Globi, protein pool, stored and reused. Porphyrin, bilirubin, excreted. Function of RBC. Transport of O2 from the lungs to the tissues. Transport of CO2 from the tissues to the lungs. Buffering action in blood. Hemoglobifugio as good buffer. By this action, it regulates the hydrogen-ion concentration, and thereby plays a role in the matarchy of acid-base balance. In blood group determination, RBC carry the blood group antigens like A antigen, B antigen, and right-hand factor. This helps in the determinatio of blood group and enables to prevent the reaction due to incompatible blood transfusion. Variations in number of RBC. Physiological variation. Increase in RBC count, polycythemia. Age, infants and children are more than adult. Sex, before puberty, after menopause. The RBC are equal to male. High altitude. In high altitude, hypoxia stimulates kidney to secrete a hormone called erythropoietin which stimulate the production of RBC. Muscular exercise. Release of adrenaline squeeze the RBC from blood. Increases of erythropoietin stimulate the bone marrow production. Emotional condition. Emotional conditions such as anxiety leads increase in the sympathetic activity and contraction of spleen. Increased environmental condition, RBC production increase. After meal, slight increase in RBC after each meal. Decrease in RBC count. High barometric pressure, where in deep sea, oxygen tension of blood is higher, the RBC count decreases. During sleep, all activities of the body are decreased during sleep. So, the RBC production also decreased. Pregnancy, because of increase in ECF volume, increase in plasma volume also resulting in hemodilutio. So there is a relative reduction in RBC. Variation in size of RBC. Microcytes, present in iron deficiency anemia. Prolonged forced breathing. Increased osmotic pressure in blood. Macrocytes, present in megaloblastic anemia. Muscular exercise. Decreased osmotic pressure in blood. Azocytosis, found in pernicious anemia. Variations in shape of RBC. Cretio, shrinkage as in hypertonic conditions. Spherocytosis, globular forms as in hypotoic conditions. Elliptocytosis, elliptical shape as in certain types of anemia. Sickle cell, crescentic shape as in sickle cell anemia. Poikilocytosis, unusual shapes due to deforming cell membrane. The shape will be of flask, hammer or any other unusual shape. Thanks for watching DENTOLOGY Notes. Please subscribe.